Okay. Good morning, everybody. Welcome to our Buckeye Neurotrauma lecture series. And the goal of this lecture is to allow our researchers here at OSU in neurotrauma and beyond to speak about their work publicly and allow everybody who wants to join us to join us. So today it's my pleasure and honor to introduce our speaker today, who is my boss, who's the chair of neuroscience, Phillip Popovich. He's also a professor and Ray Poppleton Research Designated Chair. He is the director of the Center for Brain and Spinal Cord Research and the executive director of the Belford Center for Spinal Cord Injury. Phil did his BS at Penn State, followed by a PhD at OSU, where he focused on spinal cord injury, and then a postdoc at OSU as well in neuroimmunology. So he's taken these two things, spinal cord injury and neuroimmunology, and put them together to have a very successful research career. He has many, many honors. Two that stand out to me are the University Distinguished Scholar and an AAS Fellow. He has given almost 200 lectures worldwide. He has over 140 papers for the last time I checked, and these papers are in great journals. But more than that, if you look at the impact they have on the field, it's tremendous. So very impactful work from the Popovich Lab. And he's funded by a NINDS R35, an SCI training program, and also the co-director of a T32 in neuroimmunology, which I know we're very proud of here at OSU. So I can't say enough about Phil's great research and his leadership abilities and what he's meant to me in my career, but I'm very happy he's here and allow him to talk about his most recent work, Spinal Cord Injury Causes an Acquired Bone Marrow Failure Syndrome. So thank you, Phil.
Thanks a lot, John. Can you hear me okay? Okay, great. Welcome, everybody. Good morning. Thanks for coming to our Buckeye Neurotrauma lectures. This is, as John said, this is our second. John let us off about a month ago, and I'm honored to be our second speaker. And I thought I'd try following in John's footsteps and giving you a new lecture. I've never given this talk before. It's just recently created. It's on data that we published, mostly on data that we published near the end of last year. And so I'm looking forward to sharing it with you and getting any feedback that you may have about the project. Next slide is really a requirement to give you my speaker disclosures, and I won't dwell on that. We'll move on. So I'm going to talk to you today about traumatic spinal cord injuries. And I use this slide really to just bring everybody, ground everybody to the fact that unlike other lectures you may hear about neurological diseases that have different susceptibility traits or different etiologies, spinal cord injury can happen to any of us. These are accidents. Just living your everyday life, you can be placed in a situation where you are at risk for contracting a spinal cord injury. Regardless of how this injury occurs, how it happens to you, the end result is devastating and permanent, typically resulting in severe and lasting neurological impairment, which visually manifests as inability to move, paralysis. And so this symbol here, this is the universal symbol for disability. And when we see or meet somebody with a spinal cord injury, this is typically how we first meet them, in a chair. And if you're not in the field of spinal cord injury, if you don't have experience with people with spinal cord injury, you are really, it really becomes very evident to you that they can't move very well. Their legs don't move. Sometimes their hands don't move, depending on the level and severity of the injury. And so we're under this perception that spinal cord injury is really a big problem of paralysis. And in fact, it is, but it's not the most significant problem that many of these people deal with on a regular basis. It's a host of other problems that are more debilitating in terms of their daily life. So spinal cord injury really is a syndrome of neurological complications or comorbidities. And this list here, this bulleted list, is many of the most pressing complications of spinal cord injury that people living with a spinal cord injury will talk about as being the things that they want us as researchers to work on. And so many of these bullet points are topics of interest and research projects within the Belford Center for Spinal Cord Injury that myself, my colleagues, Dana McTighe and Jan Schwab, are working on every day. And the one that I've outlined in boldface here, the immune dysfunction, is really a big topic in my laboratory and is also one of the focus areas within the Belford Center. And it's really going to be the primary point of discussion today. So why should we care about immune dysfunction? Well, it's important, obviously, to have a functional immune system because it's the immune system that keeps us safe from infections. So we all need a functional immune system. But people with spinal cord injury have a dysfunctional immune system. And this dysfunctional immune system has a lot of different implications for their health and well-being and their overall survival. Just to illustrate this point, I want to show you some data that was produced by Marcel Kamp and Jan Schwab a few years ago. And what this slide illustrates is that if you analyze data from spinal injured patients, these are high-level severe spinal cord injured patients, and you follow these people over 10 years, what you see is the individuals who contracted an infection, whether it was pneumonia or a wound infection within the first year post-injury, their survival rate over the next 10 years is decreased relative to individuals who do not contract an infection within the first year. It's about a 30+% difference in survival rate. So this tells us that an impaired immune system leading to increased infectious susceptibility has implications for overall survival and morbidity over the length of the individual's lifetime. So it's a pretty important topic that we need to understand more about.
Hey, Phil, I think your mic might be scratched against your shirt. I'm hearing a little feedback there. I don't know if other people are hearing it, but it sounds like a little scratchy.
Let me put it up like that and see. Does that fix it?
That sounds way better, yeah.
All right, thanks for letting me know, John.
Yeah.
So why does injury increase susceptibility to infection, and why are individuals more prone to infectious complications, specifically those that require rehospitalization? There's a lot of different reasons for it, but let's look at the root cause. We believe the root cause is basically a break in homeostasis and crosstalk between the nervous and the immune system. The spinal cord is actually the nexus for all communication between the body and the immune organs. The information comes back as afferent signaling in a variety of different ways, travels through the brain and the brainstem, and then the information converges on presympathetic neurons, which then will travel down the spinal cord to distribute information to sympathetic preganglionic neurons that then connect all immune organs throughout the body. And so a spinal cord injury breaks this neuro-immune homeostasis. And a number of different projects in our lab have tackled immunological dysfunction at the level of some of these different immune organs. What we're going to focus on today is how injury affects the bone marrow. So why care about the bone marrow? Why focus on the bone marrow? The answer is pretty simple, actually. The bone marrow, unlike other immune organs, this is our primary generative lymphoid tissue. This is where all blood cells, red blood cells, white blood cells, are born. All right, so in order to seed the immunological organs throughout our body and in our tissues, you need a functional and homeostatic bone marrow environment. And you need, within the bone marrow environment, to have healthy and functional hematopoietic blood and blood stem cells. It's important that they're healthy and functional because hematopoiesis is a key event for fueling the blood cells throughout our body. And the hematopoiesis occurs mostly in the bone marrow. It's an essential process for host defense and tissue repair. And again, our mature immune cells, the immune cells that seed our secondary lymphoid organs, like our lymph nodes and our spleen, develop from bone marrow hematopoietic stem and progenitor cells in the bone marrow. And we also know, and again, in the context of spinal cord injury, that the proliferation, differentiation, retention, and release of HSPCs from bone marrow is controlled by hormones and the sympathetic nervous system. So it's a pretty obvious organ to study, which is surprising given that really hasn't been that much work done on it. There is some precedent, however. There's at least 2 papers in the clinical literature, and these date back about 15 to 20 years. And if you'll notice from the data here on the right, you can see that we're talking about very small patient cohorts, 2 to 3 groups, comparative studies with group sizes anywhere between 4 and 6 patients, totals of 18 or 16. The other thing to point out about these are studies done on people 7 to 40 years post-injury or 8 months to 5 years post-injury. So fairly chronic, especially in the 40-year post-injury cohorts. But what these papers both show is that after a spinal cord injury, there are chronic or lasting deficits in bone marrow stem cell function. Now, to me, somebody who was interested in immunological consequences of spinal cord injury, I found it fascinating that these papers have been around for more than 2 decades, especially the one on the top. But there's been very little work done on trying to understand, why does this happen? How prevalent is it? What are the mechanisms that control these impaired functions? And what are the lasting implications on immune function? Are they related? So this is where I was fortunate to be able to recruit Randall Carpenter to my laboratory a number of years ago. Randall, the bulk of his PhD thesis work was focused on this problem of understanding spinal cord injury effects on the bone marrow. And the paper that you see below is really the culmination of the bulk of his dissertation work. And this was all work that was done in collaboration with Dr. Adrian Dorrance's lab and contributed to by a number of individuals. You can see all the names listed here. Some are from my lab, some are from Adrian's lab. I'll point out in particular, Mal Karanasiri, because he spent a lot of time with Randall helping us to learn about hematopoiesis. So one of the first things that Randall did when we were designing experiments to try to understand injury effects on bone marrow was just ask the basic question about proliferation. And we were asking about proliferation in the bone marrows because the process of hematopoiesis requires that these stem and progenitor cells proliferate and then differentiate. So this is sort of a checkpoint, biological checkpoint, that if it's altered, we know that there's a potential for a change in function and maybe some type of dysfunction. So Randall just asked a basic question: how does injury affect cell proliferation in bone marrow? Does it reduce it? Does it affect trafficking to secondary lymphoid organs? And the way he went about answering this question was using a novel transgenic mouse, which is pictured here on the left. This is a mitoluciferase transgenic mouse, in which dividing cells will express the luciferase reporter, and then you can image it in vivo using the NIVIS imaging system. And what you're looking at on the top are sham-injured control animals, pre-injury, and then 7 days post-surgery, pre-surgery and 7 days post-surgery. And on the bottom, spinal cord injured animals, pre and then 7 days post-injury. What you're looking at, here, let me use the pointer to illustrate this. Here, in these elliptical regions, these are the femur and tibias that have been outlined to analyze the relative level of luciferase. And if you just compare images here to images here, you can begin to appreciate that the magnitude, the intensity of the signal is increased after spinal cord injury. And this is corresponding with an increased luminescence, which is indicative of an increased proliferation. And if you track this over time in the same animals, you begin to see that there's a change. There's a significant increase that reduces somewhat over time, but stays elevated above sham-injured animals, even out to a month post-injury. So there's a hyperproliferative response in the long bones of these mice below the level of the injury between 3 and 7 days post-injury. Now, given that the peak response was prevalent within the first 3 days post-injury, this is where Randall focused the majority of the study. So all the data that I'm going to show you from this point on was obtained at 3 days post-injury, which is at the peak of this proliferative response. So Randall was able to prove that the cell proliferation is increasing after injury, and it's increasing predominantly within this first week post-injury. But using the mitoluciferase transgenic mouse, we really can't tell which cells are proliferating. And again, I told you that the process of hematopoiesis requires that the hematopoietic stem and progenitor cells are the cells that proliferate. So how do we prove that the HSPCs are in fact the cells contributing to this hyperproliferative signal? In order to do that, you really need to label the cells. And for those of you who are not familiar with hematopoietic schematics like this, I'm going to walk you through this. I certainly wasn't when we started, and I'm still learning myself. But you can see based on this, this is a canonical picture you'd see in any textbook describing hematopoiesis, that the process of hematopoiesis starts with pluripotent stem cells. that then give rise over time to more differentiated stem cells. They're still pluripotent and then ultimately multipotent before they mature into white blood cells and red blood cells. And if this is the cell population we're interested in studying, we need to be able to apply different panels of antibodies to distinguish them from other lineages and mature blood cells. So what Randall did was develop antibody panels in collaboration with the Dorrance lab that allowed us to label lineage negative, SCA1 positive, c-KIT positive HSPCs, also referred to as LSK cells. So you'll see me show that interchangeably on slides moving forward, and I may refer to them as LSK or HSPCs. So Randall labeled cells during that hyperproliferative period using these antibody panels and also applied a nuclear dye so that we could track the cell cycle. And when he did that and evaluated it in the context of spinal cord injury, he noted that there was a significant increase in proliferation, specifically within the LSK population. Okay, and here you can see the data quantified and mean across the group. And here's an example of a flow plot showing you the increase in proliferative response in the SG2 to M phase of the cell cycle. The other interesting thing that Randall noted is that if he extended this analysis of the total number of LSK cells over time post-injury, he saw that the hyperproliferative response at 3 days ultimately led to an accumulation of stem progenitor cells within the bone marrow at 1 week and also at 1 month post-injury. So you can see there's this gradual increase in time, over time, in the total number of stem and progenitor cells that accumulated in the bone marrow. So the cells are proliferating, hyperproliferative, and they're accumulating in the bone marrow. This implies that they're actually not getting out, and there's some type of imbalance or break in the homeostasis. Typically, when HSPCs proliferate, there's signals that drive the release of these cells so that they can go out into the circulation, help seed different organs, and develop a mature immune system. In this case, in this context, you see that they're proliferating, but they're accumulating. They're not leaving the bone marrow. And you can appreciate that a little bit better here in this image, where you can see if we image the long bone, and this is showing you examples from 4 separate mice, and you can just see the cellular density, with red being the most dense concentration of cells. You can see that the sham injured animals are quite different from the long bones and spinal cord injured animals. So there's just more cells accumulating, more stem cells accumulating in the bone marrow. Are they in fact being sequestered? Really, to prove that definitively, in addition to the data I've shown you, you need to go out into the periphery and look for the presence of these HSPCs or these LSK cells. So Randall did that. He looked specifically within the blood and a secondary lymphoid organ, the spleen. He used an assay that allows him to look specifically for HSPCs, and this is called a methocold assay, where you can actually isolate the tissue or the blood. You place it into a culture condition that is conducive for the growth and the proliferation and the differentiation of the stem cells, and you can actually count the new number of colonies that are forming as a result of the presence of the HSPC. And you can see that in the blood and in the spleen after spinal cord injury, there's just not that many HSPCs around. There's significant impairment relative to sham injured controls. So these cells are in fact accumulating in the bone marrow. They're proliferating a lot, but they're just, something is causing them to stick, not be able to get out of the bone marrow into the secondary lymphoid tissues or the blood. So you would expect that as a result of this, there would be an impairment in the total number of mature white blood cells that accumulate in secondary lymphoid tissues and in the blood. So again, if we just look at this slide, you can again see, right, if these cells are becoming trapped, they can't give rise to this second order LK cells, which ultimately give rise to the mature immune cells. So what Randall did to try to prove this lack of differentiation and the absence of maturation in the periphery was look for mature leukocyte markers in the periphery and quantify it. And the next slide shows that, in fact, after a spinal cord injury, within the first 3 days, you can see there's a significant decrease in total white blood cells in the blood. It's primarily caused by a deficit in the maturation of lymphocytes, T and B lymphocytes. So there's an acute lymphopenia. Note that innate immune cells, both neutrophils and monocytes, don't seem to be as robustly affected as lymphocytes, and I'll come back to that. But ultimately, what these changes do is within the first 3 days post-injury, they shift the immunological potential towards more of an innate immune response, but it's an innate immune response that's still impaired, okay? All right, so far what I've told you is that injury causes hyperproliferative response of stem and progenitor cells in the bone marrow, leading to a hypercellular response. And these cells are being retained in the bone marrow. They're not seeding secondary lymphoid organs, and they're not maturing into white blood cells in the blood. So next, Randall asked, what are the mechanisms controlling this sequestration phenomena? Why are cells being stuck in the bone marrow? And this was kind of a daunting next step in the project because there's really a lot of different things that could be going on. But we let the literature lead us to help us develop specific hypotheses. And one of an active area of research is trying to understand the relationship between the bone marrow stromal cell population and the HSPCs and mature white blood cells within the marrow compartment. And we know from the literature that there's a number of different stimuli that regulate in a tonic manner signals that are originating from the stromal cells. And those signals communicate directly with the stem progenitor cells and the mature white blood cells in the bone marrow. One of them is cytokine and chemokine signaling. So under normal circumstances, as the stromal cells are releasing cytokines and chemokines, that interacts with specific receptors on the stem and progenitor cells. And if the signal is present, what happens is that it causes the HSPC to stay within the bone marrow compartment. When the signal disappears and the chemokines are no longer expressed, the signal is released and the cells will enter into the blood. So based on that simple schematic and simple scenario that I just described, we would predict that after a spinal cord injury, there is an excess of signaling, causing excess triggering of a retention phenomena within the bone marrow. So how do we study that? Well, what Randall did was really a simple experiment. At first, he went in and he said, the literature tells me there's many different factors that contribute to this signaling, there's many different cytokines and chemokines. Why don't I just analyze that in the bone marrow compartment? Here's a list of 6 that he looked at. And of these, 5 of them were significantly elevated after a spinal cord injury. Notably, 2 of them, CXCL12, the ligand, and CXCR4, the receptor, were upregulated after a spinal cord injury. And this ligand receptor pair are well known to be involved in bone marrow stromal cell, hematopoietic stem and progenitor cell retention. And so the fact that they were elevated was a potential candidate for a mechanism that might be disrupted after a spinal cord injury. Randall did a little bit more digging. He isolated the extracellular fraction, extracellular fluid within the bone marrow to specifically look and quantify the amount of CXCL12 protein. And again, you can see that after injury, it's significantly elevated. And he also isolated the stem and progenitor cells and using flow cytometry, analyzed the relative level of expression of the receptor for CXCL12, and that's CXCR4. And again, you can see that in injured animals, the receptor is highly upregulated on the stem and progenitor cell population. So this is a likely candidate that might help explain the retention phenomena. There's excess signaling through this ligand receptor pair. Now, to really prove a causal relationship in the retention phenomena, you need to try to disrupt this signaling pathway. And fortunately, there's actually a really well-used drug, well-known drug, that's used clinically in many bone marrow cancers to mobilize white blood cells from the bone marrow. This is a drug called Mozabil or plerixafor, oftentimes referred to as AMD 3100 as well. Again, it's an FDA-approved immune stimulant that works by antagonizing the CXCR4 receptor. So Randall just asked, can this drug reverse the spinal cord injury-induced bone marrow failure? And what he did was simply inject animals post-injury with the drug with the goal of blocking CXCR4 to see if it could reverse the phenomena that he had observed to this point. And these data show that it is actually a very effective, consistently effective and robust treatment for reversal of the acute bone marrow failure. On the top, which you're looking at, are these methylcalled assays that I described a few slides ago. You can see on the right, these are images of individual cultures from a spinal cord injured animal that was treated with vehicle. You can see there's no colonies forming, just one here. But the animals that got injected with AMD 3100, there's a lot of colonies forming in cultures developed from the blood. So there's a significant increase in blood CFCs, indicating that the HSPCs, or the stem progenitor cells, are being mobilized from the bone marrow of spinal cord injured animals when they get plerixafor injections. This ultimately leads to an increase in circulating white blood cells and a reversal of the acute lymphopenia that I described to you earlier. Note that neutrophils and monocytes are also significantly increased in the blood. So it does seem to be that the AMD 3100 injections are overcoming whatever spinal cord injury is doing to the bone marrow compartment to cause the retention phenomena and the acute onset of immune dysfunction. So what are the long-term immunological consequences of this failure? So everything I've described to this point has been characterized within the first 3 days post-injury. And you can begin to guess, that maybe there'll be long-term consequences, or you might predict that there'll be a reversal. With time, this may wear off and things may get back to normal. But keep in mind, early on, I told you that those two clinical papers, dating 15 to 20 years ago, we're looking at patients that are up to 40 years post-injury that still showed deficits in bone marrow stem cell function. So it's likely that the consequences of this acute acquired bone marrow failure are long-lasting, leading into and maybe being responsible for the long-term immunological dysfunction that is present in people with spinal cord injury. So again, how do we study that? How do we begin to interrogate the system in a way to test whether or not there's long-term consequences associated with this acute onset failure? The way Randall did it was as follows. So this is the schematic overview of his experiment. He compared sham injured animals again to spinal cord injured animals. He waited about 6 to 8 weeks post-injury, and then he injected the animals with systemic endotoxin. So why endotoxin? Because it is a highly reproducible and potent immune stimulus. It also challenges directly bone marrow hematopoiesis. It triggers a hematopoietic response and mobilization of not only stem and progenitor cells, but also mature white blood cells, causing them to leave the bone marrow, enter into the circulation to, again, attempt to resolve the immune stimulation caused by endotoxin. So he injected them daily for 3 days, a single injection, and then waited 2 to 14 days post-LPS injection, during which time he was analyzing them using the myeloluciferase transgenic mice, again, to look at proliferation in the bone marrow, and then end stage looking at total white blood cell counts, as well as using the methylcalled assay to look for the presence of the HSPCs, functional presence of the HSPCs. So again, on the left, you're looking at myeloluciferase transgenic mice, and you're looking at the effects of injury pre-LPS, okay? And then 11 days post-LPS. Just focus on what's happening here and here. So you can see that, again, the LPS response triggered a potent response. You don't see as much of a response in the sham injured animals, although it is increased. But this hyperproliferative response, if you track it over time, continues to rise. So just like we saw early, this hyperproliferative response within 3 to 7 days post-injury in the absence of any kind of immune stimulus, even several weeks or months post-injury, it's still present. And there doesn't seem to be any attempt to regulate the response in the animals with spinal cord injury. When you look at the presence of the hematopoietic stem and progenitor cells in the blood, again, you can see that this hyperproliferative response is associated with a decrease in release of these stem and progenitor cells into the circulation. It's even still present at 11 days post-LPS. So this is a lasting response. It's a lasting impairment in mobilization and surveillance by immune cells. And at the level of the mature white blood cells, you can see, again, decrease in white blood cells compared to sham injured group. Again, it's predominantly caused by lymphopenia, the inability to produce a mature lymphocyte population, but not so much by a deficit in innate immune cell responses. So inflammatory stimulus fails to mobilize these cells and produce mature lymphocyte responses in chronic spinal cord injury mice. So this is a lasting deficit. So Randall, there was a couple other experiments in Randall's thesis that I'm not going to go into today. Again, it's in that paper that I showed you early on. His project was fantastic. It raised so many interesting questions that we had not had an opportunity to follow up on since he left the laboratory. But I was fortunate enough to recruit Kylie DePetro, who's a new MD PhD student in the lab. And she, together with a postdoc from Adrian Dorrance's lab, Rohan Kulkarni, are beginning to follow up on these studies. And here's just a slide illustrating some of the ongoing studies and questions that we're interested in answering in collaboration. And I'm just going to give you, in the next few minutes here, just a preview of some of the work that Kylie's been doing to follow up on Randall's work. So as a reminder, everything that I just talked to you about related to Randall's thesis work was focused on this 3-day time point, okay? But how early does this response happen? How early does the onset of a drop in white blood cells and the onset of lymphopenia occur? And what are the mechanisms that may cause these changes? What Kylie's done is really replicate Randall's studies, but move the experiments to 1 day post-injury. She moved it up to 1 day. And why did we move it up to 1 day post-injury? Let me go back quickly. If you look at this slide, you can actually see that the proliferative response is starting to increase at 1 day post-injury. It's certainly maximal at 3 days, but it's going up here. So this is one of the reasons why we backed it up a little bit, to try to find out how quickly does this injury-induced change occur. So again, Kylie just repeated Randall's experiments. And when she did this, what she found, she basically was able to replicate the acute onset reduction in white blood cells. And again, you can see this profound lymphopenia. This is all within 24 hours. But the interesting thing that Kylie found was that there was also a deficiency in the number of mature neutrophils and monocytes in the circulation. So unlike Randall's data at 3 days, there's an impairment in mobilization and maturation of innate immune cells as well. So something's happening between 1 and 3 days that is compensating for this acute deficit. And it only seems to be preferentially affecting innate immune cells. What is that? Well, it's possible that injury is influencing stem and progenitor cell populations a little bit farther down on the lymphoid progenitor and common myeloid progenitor lineage side. So it's certainly affecting these cells, but is it imprinting some kind of signal in these cells that's differentially affecting these different progenitor cell populations that ultimately give rise to innate immune cells versus lymphoid cells? Now, Randall did do some work on this to try to get some insight to this. So again, what he had to do was use a variety of different antibodies to look at these different progenitor cell populations, as well as some of the myeloid precursor populations, using, again, different antibody panels. And what he was able to find when he did that was that basically all of the different unique progenitor cell populations that he looked at were significantly increased by injury. This one right here, the GMP, corresponds with this cell population here. Note that Randall did not look at how injury affects the common lymphoid progenitor population. But Kylie has started to look at that together with Rohan. And what they found is that just like Randall saw at 3 days, there's an increase in the granulocyte monocyte precursor cells, but there's a decrease in the common lymphocyte progenitors. Again, this goes hand in hand with what we're seeing at the level of mature white blood cells. The innate immune cells are increasing, but there's a profound lymphopenia that seems to be lasting and cannot be reversed even with an immune stimulation at later times post-injury. So this is one of the exciting areas I think we're going to pursue. Why is there a preferential effect of injury on one lineage of cells versus the other? How early does it happen? Is it reversible? Is this a mechanism that's underlying chronic immune suppression in spinal cord injured animals and people with spinal cord injury? So this is a summary of everything I've talked to you about today. I don't think I need to go back through it. I think that I just want you to see it visually. to appreciate the fact that we've made some progress. We certainly don't have all the answers at this point. The one thing I can tell you for sure that we know in terms of an intervention is that plerixafor is very potent at reversing the acute onset bone marrow failure that's due to the CXCL12, CXCR4 signaling pathway. Whether or not that will overcome chronic immune dysfunction, we don't know. We haven't actually analyzed that. We have done a couple of experiments that indicate that it's not enough to overcome chronic immune dysfunction. It does mobilize cells and it increases the number of mature lymphocytes, but things still aren't normal in an animal with spinal cord injury. So a lot more work to be done there. And my hope is that with the help of our great collaborators in the Dorrance laboratory, many fantastic investigators, past and present in my laboratory, and through Kylie's dissertation work, that we'll be able to get some answers to that. So I wanted to end by thanking this entire team. Thanks to Randall. Hopefully, I'm not sure if he's on the seminar today or not, but it would be great to hear from him if he is. And also from the support from a variety of different foundations, the NIH and the Belford Family Charitable Trust. With that, I'll end and answer any questions that you have. Thanks very much for attending, and I hope to hear from you.
Great. Thanks, Phil.